What are the technologies used for PEG conjugation by a chemical point of view? One of the first method used for protein conjugation was the reaction of activated PEG to the amino groups of the protein. This is due to the fact that the amino groups of the proteins are very abundant in the protein structures, and they are exposed on the surface, and they are rather well reactive. And so it is rather easy to conjugate polymers or to these amino groups. However, throughout the years, several other methods have been developed to obtain more selective conjugation and to exploit also different functional groups of the proteins. For example, the conjugation to disulfide bonds, and we will see later an example, the conjugation of specific amino acids, and also the conjugation to thiol groups. So we can see here the conjugation to the PEG and several different derivatives. For example, the aldehyde groups is well used to conjugate the terminal amino groups of proteins. This is rather selective by working at specific pHs. Which make these amino groups more reactive than the lysine amino groups of the proteins. Then there is the isocyanate, the phenylglyoxal, and then there are several derivatives which have been used for conjugation to the thiol groups. Conjugation of thiol groups is very interesting because thiol groups are very seldom in the protein structure, and usually they are engaged in disulfide bonds because they are exploited by proteins to stabilize the protein. But it is very easy to obtain derivatives of proteins by genetic engineering, introducing cysteine in sites of the proteins which are far from the active site of the proteins, and then it is possible to conjugate selectively these thiol groups by using specifically activated polyethylene glycols. So it is possible to have a site-directed conjugation, and this allows for the obtaining of derivatives with very well defined structure. And finally, it is reported this very interesting method for conjugation of PEG to the disulfide bond, which was developed by Steve Brocchini a few years ago. So the other methods to obtain selective conjugation is the use of enzymatic conjugation, and there are two methods which have been particularly investigated. The first one is based on transglutaminase. Transglutaminase allows for the conjugation to amino groups, to glutamine groups, and so it is possible to use a PEG terminating with an amino group to conjugate to the glutamine functional groups of proteins or vice versa. And this is very interesting because enzymes are very specific in the substrate of conjugation. Another method was developed a few years ago in 2006 by the FIRS, and is PEGylation, and this is a multi-step conjugation which exploits different enzymes. And the first one, the first step is the galactosylation of sites which have not been engaged in the glycosylation naturally, and for example, serine or threonine. And then there is the conjugation of PEG through a sialylated PEG through a sialyl transferase. And also in this case, it's possible to obtain derivatives with a selected site of PEGylation. So in this slide, I will show you the example of interferon that I mentioned before. This is very interesting because are two different strategies to obtain derivatives with a very high therapeutic activity. Interferon alpha 2b and interferon alpha 2a. In the first case, in the case of interferon alpha 2b, we have the conjugation of PEG 12 kilodalton to the amino groups of the protein, and one of the conjugation was obtained at the histidine 34 of the sequence of this cytokine. And these derivatives is very interesting because the bond is cleavable, and so it is the result is that the PEG is released throughout the time after administration, and the native protein is released. So there is not the problem of high hindrance of the polymers, which a reduction of the activity by masking of the active site. Indeed, these derivatives maintains 35, more or less 35% of the activity of the native protein. And this is due to the fact that the molecular size of the polymer is only 12 kilodalton, is a linear polymer. Only one polymer chain is attached to the protein, even though in a random way, but only one polymer per protein molecule, and nearly 50% of the polymer is attached to histidine 34 by obtaining a releasable PEG. On the other side, we have interferon alpha 2a. In such a case, a 40 kilodalton PEG was conjugated to the protein. The bond was stable, so we didn't have a releasable PEG. The high molecular weight and the branch shape of the polymers produce a derivative with only 7% of activity, as I said before. So the two derivatives are both excellent therapeutic agents. They have been used for, and they are still used in therapy with high activity and excellent performance, even though they are different in terms of activity, but they have also differences in terms of permanence in the bloodstream. So in the conjugation method affect the protein properties, and in particular, they affect the permanence in the bloodstream and the immunogenicity of the protein, and also the stability. Usually, only PEG is activated only on one side to obtain mono-PEGylation. So that means that one PEG chain is attached to the protein. When the PEG is activated to both sides of the chain, we obtain a bifunctional PEG with the possibility to obtain cross-linked bioconjugate, like in the PEG unigalsidases alpha. Then we have the different conjugation method, and we can obtain single one only polymer per protein, such as in the case of the interferons, or we can have several polymer chains attached to the proteins, like in PEG asparaginase or PEG uricase or PEG adenosine deaminase. And in both cases, the conjugation is random, so the PEG is conjugated to different sites of the proteins, and the activity of the protein is an average of the activity of all the isomers which are obtained. Alternatively, there is the site-directed conjugation by using different strategies like the cysteine conjugation or the conjugation to the terminal amino groups, or by using enzymes. So in this slide, we can see the effect of the PEG mass of the conjugate on the permanence in the bloodstream. And as an example of avidin, which was conjugated with a PEG 5 kilodalton, PEG 10 kilodalton, or 20 kilodalton. All these PEGs are linear chains. And we can see that by increasing the molecular weight of PEG, there is a significant, remarkable increase of the permanence in the bloodstream. On the other side, there is an example of the use of PEG in comparison with different polymers which were used throughout the years as alternatives of PEG, such as polyvinyl pyrrolidone or polyacryloyl morpholine, and also the results obtained with a linear PEG or a branched PEG. And here is reported the immunogenicity of uricase, which is an heterologous protein with high immunological properties, immunogenicity. What we can see here is that actually polyvinyl pyrrolidone and polyacryloyl morpholine are not very effective in reducing the production of anti-uricase IgM and anti-uricase IgG. And in both of them are not very protective, and they do not reduce very much the immunogenicity of this protein. On the other side, PEG, linear PEG, is much more effective in reducing both IgM and IgG, but the best performance was obtained with a branched PEG, because branched PEG has the ability to cover the protein surface like an umbrella, protecting the protein inside from the recognition of the immune system, from antibodies, anti-protein, and also from enzymes and from degradation.